transmitted disease. Okay, sexually transmitted diseases, or you can call them sexually transmitted infections. So for the sexually transmitted infection, you need male and a female, or two males or two females, whatever. But there should be a sexual contact is must. Okay. So you will have these two lectures later on. But today we are going to. It's divided in two parts. Lecture 1, lab 1. And similarly for this there is a lab 1 and lab 2. Okay, for sexually transmitted infection. Here today I am going to cover. Dr. Father took a lab,
Yes,
lab 1.
Yeah.
So which method you use today?
Try
And what?
Yeah.
So,
you want to.
I will take care of now. We will talk about today disease of gonorrhea. Okay. One, number 2. This is my repeat. Medium, and number 3. Chlamydia. In brief, general, just actually you will have more information in the lecture. These two, Trichonema and Chlamydia. This is present here. This is Trichonema palladium, spirochete, and it is trichomatis. Okay, both these microorganisms we cannot culture them in the laboratory. Till now, for Trichonema, it's now around 80 years. We couldn't find a medium how we can grow this microbe. So we need something living cell, okay? We need something living cell, like rabbit or mice. For gonorrhea, you can culture this in the laboratory, you will see now. The purpose of this today's talk about the diagnosis, only diagnosis. And I will talk about a little bit about morphology also. Most important is its physical manifestation, its pathogenesis, it's in detail the structure of this bacteria cell wall, why they are not grown outside in the artificial media, why grown in the, we need the living cell, everything will be covered up in the lecture. Okay? Name of the scientist. Listeria, Albert Listeria was a scientist, and when he found this microbe, he gave his own name. Gonorrhea is name was a bacteria. And when you see the morphology of this bacteria, Diplococcus, have you seen the coffee ball? Coffee beans are like this, right? So this bacteria, it looks like a coffee. You see two together. Diplococcus, two cocci. Cocci is the word cocci. Cocci is that? Come on, cocci is that?
Yeah, like this.
Cocci is the shape of bacteria. The shape is cocci. Two cocci are like this, but when you see it looks like a coffee bean. It looks like a coffee bean. So this is about under the microscope. When we do a grain stain, and we see it's a gram negative bacteria, Diplococcus, and it looks like the coffee bean. It's a morphology, very simple. And for the sample, what you need, purulent discharge. Some from the bloody parts and or pus-like structure, you have to take it as a sample, because I said it is a STS, actually transmitted infection.
Yes, yes.
So actually, these infections are usually more common in young and active people, mostly affected European and American countries, okay? Because they thought it is, you know, that blah blah blah. So these are very young and the high incidence rate in college and universities. Okay? And other places also.
Just one way to transmit by sexual intercourse.
Sexually transmitted infections. Sexually transmitted. Actually, this was the hypothesis for this microbe. It was said that it attaches the sperm and sperm carries it. It was the first hypothesis. First hypothesis was made by the academicians and the scientists. But later on it was reversed. There were some other factors to carry it. Okay? That they, because till then there was not much known about this bacteria. So it was said sperm usually carries it. But later on it was found there are some other reasons. You will find it with other reasons. So for this microbe isolation in the laboratory, we can use the culture media. We can use the culture media. So here for microscope and for sampling, no questions, right? Next. Culture. You can culture this microbe on chocolate agar. Why not on the blood agar? Because the presence of the fatty acids in the blood agar, it inhibits its growth. So this microbe needs special repairments, and we usually use. What's the chocolate agar, by the way? What's chocolate agar? You need to heat it for 10 to 15 minutes. Okay? If you the chocolate, what chocolate you do? Chocolate you will eat. Yes. Very simple, you stop it. And you put it here. You put it in a factory. Okay? Then you took the sample from the affected part and it is transported to the laboratory and you culture it on the Petri plate. This is about why it becomes chocolate color, because due to the heating, red blood cells are destroyed and they are broken down and they release their components. This is one thing you can grow this bacteria, okay? Another is a Thayer-Martin medium. It was the name of a person, Martin. He synthesizes, produces a media, culture media for this bacteria. It's the enriched selective media, and he put antibiotics. Okay. He had the antibiotic in the medium. While preparing the medium, he put the antibiotics. Why?
No, floor.
Prevent growth of other Neisseria gonorrhea, because there are two types, Melanomax and other, and normal flora. So that you get particularly this microbe on the culture plate, more specific, selective media. Selective media. When we say that you use selective, for example, when you, because many bacteria you can grow it on the chocolate agar. Okay. Now you want to make sure, because when we talk about the sexual infections, sexually transmitted infections, so report should be very accurate. Because even a false positive is a big problem. That you can put a patient on a therapy of antibiotics is wastage of time, waste of money economy, and also patient suffering. So we should be very cautious while identifying these type of microorganisms. So we do one more test, Martin medium we use there, Martin. Why? Because it favors strongly the growth of Neisseria gonorrhea. Okay? Correct?
Doctor, what's mean enriched medium?
What?
Enriched medium.
And enrichment. Very simple word. Enrich something. It's a, it's a blood. When it's a blood. Okay? When you make the lysis of the blood, it releases iron, it releases protein. So this makes the enrichment of the medium. Because the same thing in the blood agar, it's not good. This microbe, it cannot grow because of the difficulty of the presence of fatty acid matter. But while the chocolate agar, same blood agar. This means when we put the blood on the heating, it breaks down, releases iron and protein, and it becomes accessible to the bacteria. It's called enrichment medium. Enrichment can be done by a number of methods.
Yes, heated the blood for 2 minutes after chocolate, then antibodies to suppress normal flora. Then doctor.
No, it's another medium. One medium is another. Hussein, where are you? First is chocolate agar, okay?
Yes.
In the chocolate agar, the bacteria can be grown, okay? Another media is a Thayer-Martin. Another media, second type of media. It's a selective media. Why? Because antibiotics are added to it, like vancomycin and other 2-3 antibiotics mixed together and put it while making the media, and it favors the growth of this bacteria. Fine? Any questions?
Doctor, are they still using chocolate?
We do, in the laboratory, in the chocolate, you have seen the growth. You are suspecting, oh, this patient is having the gonorrhea infection. it's a sexual infection.
Yes.
Immediately you do this another test to make it sure.
Yes.
To make sure. Chocolate we are using. And usually we do number of tests as much as we can to confirm these type of infections. Okay?
Okay.
Understood?
Yes.
Shall we go to next?
Yes.
Biochemical reaction. For the same microbe, gonorrhea, we can do some biochemical tests. Oxidase? What is oxidase for? Now you are in the third year. So I am sorry, you are in the second.
Addition of water
Oxidation.
No, oxidation is breaking down. You remove the oxygen. Breaking down the water.
So like
I know you forget things very
It's good.
All students, when I was a student, I also forget something. But try to reflect people like catalase positive, oxidase positive, formulase positive. What does it mean? You should remember these things. Oxidase, it's enzyme produced by the microorganism. Oxidase. What is the feature of the oxidase? If microbe is having this oxidase enzyme, it transfer, if it is present, it transfer electron to the hydrogen and it form loss to the water and energy.
Could you repeat this?
If microbe is oxidase positive, this means this bacteria is transferring the electron because this means there is presence of the cytochrome. There is presence of a cytochrome. If you remember, in our mitochondria, how we respire? Because of the cytochrome. In our mitochondria, the cytochromes transfer the electrons from cytochrome 1, cytochrome 2, cytochrome 3. In case of humans, cytochrome 4. Then finally it produces water and energy, so that our body produces energy and we can talk, we can walk. Similarly, for the bacteria, if bacteria is oxidase positive, this means there is a presence of the cytochrome. This microbe can carry the electron in presence of oxygen, and it produces the energy in the body. Enterobacteriaceae family is oxidase negative because they are anaerobes. We do not remember this. Enterobacteriaceae in the non-monobacteriaceae, all enterobacteriaceae family, they are oxidase negative. They are oxidase?
No oxygen.
So there is complete absence of this enzyme. They cannot carry the transfer of, there is no role of oxygen. There is no role of oxidase negative. But the microbe is oxidase positive. This means that microbe has a capability to carry the electron and transfer it to the presence of the oxygen and produce the. Understood what is the oxidase positive mean? This bacteria is oxidase positive. And when we see the oxidase test, this is the citric. It contains paraphenylene diamine, a chemical. That chemical is exactly same to that of this enzyme. Okay? So when we touch the bacteria on it, okay, bacteria releases this enzyme and this chemical, paraphenylene diamine, that's present in this paper citric. It acts as the cytochrome and helps in transfer of oxygen. When it reacts with the oxygen, transfer of electron organ, and you see it's blue color. Okay? Oxidase positive or usually purple color. Again, this is because of the chemical present in the Fine? Understood? Next, sugar fermentation is glucose positive. What does it mean? This means this bacteria can grow in presence of sugar in the liquid medium. We prepare a liquid medium in a test tube in a laboratory, okay? We put here sugar. And put the material inside. And next morning we will show here growth. We will show here how it can be changed in the color. But if we put another sugar, maltose, my bacteria cannot ferment it. Some bacteria can eat lactose, some bacteria can eat maltose, some can other sugars. So it depends on the bacteria. Inside its bacteria is a single cell, okay? But God has given you the preference. Somebody likes lactose, somebody do not like lactose. Famous example, non-lactose fermenter and lactose fermenter. What does that mean? Some bacteria grow on lactose, some bacteria do not grow on the lactose. It depends on the bacterial cell. So this bacteria, Neisseria gonorrhea, this bacteria, when we talk about this bacteria, it grows on sugars, it eats the sugar, glucose, correct? And if we use the another sugar, maltose, also sugar, but it will not. It will not, correct? And one is the another, this rapid test nucleic acids presence in the urine. So also we can go for the PCR reactions for this. Serological tests are not useful because when you have a lecture, you will see this microbe remain inside the cells. Okay? Okay?
Doctor, if you ask us another biochemical reaction, just mention oxidation positive.
Biochemical reaction of Neisseria gonorrhea. Biochemical test is for Neisseria gonorrhea. You will write oxidase positive, sugar fermentation, maltose negative, and glucose
What are
positive.
the principles of oxidase?
There is no, I will not ask for the principle. There is no principle. Is just you should remind actually what a reaction is. Not with this now. So first one is clear. Treponema. Treponema palladium. It is a spirochete. Spirochetes is a separate column and of the reactive. It's separate. It's a gram negative but separate them. It's there are a number of microorganisms. But one molecule is Treponema palladium. It's a gram negative bacteria, but again it has been placed in the another family. Why? Because it contains double layers. It contains two membranes. Inner membrane, outer membrane. This is the cell. This is the bacteria. There is two membranes, one and two. Usually we have in gram negative bacteria only one outer membrane, right? But in the spirochete we have two membranes, so it has been separated class. And in the spirochete there is a bacteria. It is called Treponema. It's called Treponema palladium. It causes a famous disease. What is it called?
syphilis.
Indian first prime minister died because of this disease. Okay? Because the reason I think he is recording, I cannot say what was going on. And his name was Prime Minister of India Jawaharlal Nehru. He died because of this disease. And when you had a lecture on this, it's a very dangerous disease. It remained asymptomatic for a long time, you could not notice. And it has a different stage, primary stage, secondary stage, and tertiary stage. Tertiary stage can come up 10, 20 years or 15 years later. Sometimes about 8 years later. So, about this disease, this bug is something very notorious and very dangerous bacteria. Okay, so what? No, both. Sexual transmitted infection, we could not do it. There is no way to do it in the laboratory. Number one. And number two, there is no other reservoir except the human. Only reservoir for this bacteria is a human. Reservoir. Source of this bacteria. It remains only the humans. Come from one human, go to another human. It cannot go to the monkey, it cannot go to the rabbit, it cannot go to the plant and come back just human.
Just go
Between the humans.
and then sexual.
Between the sexual contacts. Between the sexual contacts. And this is so, this bacteria again, it has been some temperature growth for around 37. Slight temperature increase outside, inactivate automatically. If we use a disinfectant, immediately this bacteria will go. But as I told you, it is a double membrane bacteria, spirochete, Treponema palladium. You will see under the microscope it is shaped like this because it has a spiral flagella inside. Between these two membranes, spiral flagella. Okay? So when we see, you see here the spiral shape. You will get that in the lecture, but here we are focusing for the lab diagrams only. So let us see in the microscopy. For syphilis, we will do this test. There are primary, secondary, and tertiary. 3 stages for this disease, okay? Primary, secondary, and tertiary. As I told you, tertiary can come later on after 5 or 10 years old. So main focus is on primary and secondary symptoms. So primary and secondary stage. In these two, and lesions occur on your, it can occur on a lip, it can occur on a tongue, it can occur on your genitals, means for the male and vagina for the female. This can come, but maybe it usually comes from the private part first. So if you, for example, somebody on a PC is having a lesion in a primary, it is called Shankry, okay? It's a painless lesion. You will see in the lecture, and you put some exudate for it, okay? You collect the exudate, transfer into the laboratory, okay? In the laboratory, it was seen under the dark field microscope, and using the fluorescent antibody stain, and it looks like this. If you use the fluorescent antibody stain, it looks like green. Fluorescent is a green. It's the reason it looks green. Or if you like this, it will look like white. Okay? Simple. In the primary stage. Primary stage usually remains for 5 weeks. Okay? Then from 7 weeks onwards up to 10 weeks, it jumps to the secondary stage. Okay? It's called secondary stage. You will find this in the lecture. Okay, inshallah. But here I am telling you, you are having a patient in your clinic, and you are suspecting that patient might have a sexually transmitted infection. When you say the past history, his pens is some structure you will see, and at that time you will recommend for the dark microscopic test. Take the sample, send it to the laboratory, it can be found. Okay? It is a direct observation. Okay? Took a sample in the laboratory under microscope.
Principal, without stain?
Dark field, yes, no, but it is not green. Here is this fluorescent antibody usually. Okay? This is very simple. If this comes picture to the exam, you will immediately write it to the treponema palladium. It's a gram-negative bacteria, finish close. Okay? Nothing more to write.
Okay, check, light what? Check.
Shankly is spondylometallida. Again, it grows. Then you see the growth, painless growth.
Any location, not only pain.
In the skin, in the penis, in the lips, on the tongue. Because when disease advances, tertiary stage, the advanced disease, it can